Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Expand Your T Cell Manufacturing Capacities in an Automated Closed System to Shorten Your Production Time. I'm Abby Pinchbeck, an editor at BioInsights. Joining me today are Sophia Lollies and Ian Johnston, who will discuss process details and differentiators, along with limitations and how to choose the right process type for your requirements. Now I'd like to introduce our presenters for today. Sophia Lollies is the Global Product Manager for the Clinical Engineered Cells portfolio, including CAR T-cell immunotherapy at Miltenyi Biotec. Since 2021, she has been working with R&D and the clinical development team to advance the development of enabling tools for CAR and TCR T-cell therapy. Before that, she supported the development of clinical applications on the CliniMACS Prodigy for industrial customers. Ian Johnston is the Senior Project Manager for R&D T-cells at Miltenyi Biotec. Since joining the team in 1999, Dr. Johnston has developed a number of reagents and procedures to improve the efficiency of genetic modification of primary cells such as human T and stem cells. In addition, Dr. Johnston has been lead development of numerous products for magnetic isolation of cells, molecules, organelles, and for use in the field of HIV research. So without any further ado, I'm going to hand over to Sophia to kick us off with today's presentation.
Thank you for the introduction. I'm going to start off our webinar, and we will talk about how to expand your T-cell manufacturing capacities in an automated closed system to shorten your production time. And first of all, I will give an introduction into our CliniMACS Prodigy Platform, a platform for an automated T-cell manufacturing. Then I will dive into the T Cell Transduction large-scale system and application on the platform and a complete workflow solution. And then I will hand over to Ian, who will give an insight into process development data on CD8-positive TCR-modified T cell engineering in AML. And after that, we will start our Q&A session. This is our disclaimer, which is mainly about the products we provide. And first of all, I would like to talk about the success story of Miltenyi Biotec's T-cell engineering manufacturing strategy. And the CliniMACS Prodigy device was launched in 2013. And after that, the success story continued, for instance, in 2018, when the first trial patient was treated with a product which was produced on the CliniMACS Prodigy in our own cell factory. And further on, for instance, in 2021, our CliniMACS electroporator and the formulation unit, so modules of the devices, were launched. And then last year, we released the CliniMACS Prodigy T Cell Transduction large-scale application, which is the application that we will talk about in this webinar. And we also released the CliniMACS Workbench. Also, we have a very established track record for clinical T-cell engineering. And here in this track record, you can nicely see in purple the preclinical data and manufacturing optimization. And here, for instance, different enrichment strategies were established, cultivation strategies, also fresh and frozen starting material was used, also different functional tests and so on. And then in orange, you can see first clinical results, such as bispecific anti-CD20 and anti-CD19 CAR T cells, or the CD19 CAR T cells in SLE, and so on. So as you nicely can see, the CliniMACS Prodigy platform is already widely used in the field and used in the preclinical as well as in the clinical field. To give you a quick introduction into the Prodigy device, I want to start by talking about the manual process that you have to perform when you want to generate engineered T cells or any other kind of engineered cell. As you nicely can see, is that you have to perform all the different steps from starting material to washing, selection, activation, gene engineering, expansion, formulation, and filling, all separately, and that are up to 20 transfers and manual handling steps. And that gives many windows for variability and errors, and also leads to increased operational and capital expenditures. So it's quite costly. And to reduce these errors and to reduce costs, we have developed the CliniMACS Prodigy Platform, which is an automated cell manufacturing platform that combines all these steps in one device. It is an end-to-end automation in a closed system. All the results are reproducible and consistent. It can reduce operator hands-on time, and no extensive training of personnel is required. To show you further how the CliniMACS Prodigy Platform is able to cover your entire clinical workflow, I would like to show you the 4 components of the platform. So it consists of the CliniMACS Prodigy instrument, so the fully automated platform for increased reproducibility and standardization. We also offer Max GMP reagents. So all our reagents we offer in GMP quality and they are validated. And then we also offer closed tubing sets and consumables to ensure a closed and safe environment. And lastly, we also have the CliniMACS Prodigy software. So we offer several applications on the device that offer a standardized process, but are also configurable and can be flexibly adjusted. Here, I just want to show you quickly some of the parts of the device. So in the orange squares, you can see the components of the Prodigy device itself. So it has a peristaltic pump and valves to control liquid flow. In the centricite unit, the cell processing and the culture takes place. We have a touch screen with a guided user interference to control the process. And then the magnet unit holds our separation column to separate the cells. We have a barcode reader to track all the consumables and a sealer to remove sample pouches and bags. Here in blue, you see our components of the CliniMACS electroporator, which is an add-on device. And here below, the CliniMACS formulation unit for the final filling of your cell product is shown. And all these components, all in all, ensure flexibility. They can reduce costs. They minimize more consistent product quality. And now I'd like to continue with our T-cell engineering workflows. So as you see here, we offer 3 different T-cell engineering workflows. The T Cell Transduction, short TCT process, which is mainly for CAR T cell generation for the treatment of liquid cancers. And this is our most used application in clinical trials. Then we also have the T cell engineering workflow, which consists of the CliniMACS electroporator. So this enables a complex gene engineering of CAR T cells or any other kind of T cell. So we enable CRISPR-Cas, knock-in, knock-outs, and so on. So we can treat diverse indications here. And lastly, we have the T cell transduction large-scale process, which is quite new, or the newest application that we offer. And the application areas here are for people interested in CAR T, but also TCR T cells for the solid tumor treatment, and any other therapies that require high cell doses. And now I'd like to give you an introduction into this application, short the TCT-LS. Here you see all the components of the T cell transduction large-scale process. So the workflow consists of the cell selection, activation, the transduction, expansion, harvest, and afterwards we also enable cell analysis, but that is not on the platform itself. We offer reagents for the cell selection, like CliniMACS CD4 and CD8 reagents. The activation of T cells is enabled by our max GMP T cell transact large scale. Expansion takes place with max GMP media and max GMP cytokines such as IL-2 or IL-7 and 15 in combination. And harvest is performed in our formulation solution. And we also have a formulation and cryo kit in case the cell product in the end needs to be cryopreserved. The tubing set is the tubing set 620, which has an enlarged cultivation chamber. And with that enlarged cultivation chamber, we can reach a final cell number of up to 20 billion T cells in 12 days. Here, in comparison to our TCT process, so in the TCT process, we are able to generate up to 6.5 E9 T cells. And here you can see the cultivation chamber of the TS520 tubing set that's used in this application. In the TCT-LS, we have an enlarged cultivation chamber that you can see here. And with that, we enable, as I just said, the generation of up to 20 billion T cells, which is quite more. Just some fast facts about the process capacity and the specifications. So the starting material for enrichment, the process allows fresh or frozen leukapheresis cell products. And here we allow up to 10 times 10 to the 9th white blood cells. And in this leukapheresis, we allow up to 3 times 10 to the 9 labeled cells, meaning T cells. And here, depending on whether we start with fresh or with frozen material, we allow different volumes. Fresh, we allow between 50 to 600 milliliters. For frozen material, we allow 50 to 300 milliliters. This is due to the automated dilution that is performed of the frozen material or the thawed material inside the system. And then the starting material for culture, here we recommend to seed 4 times 10 to the 8 enriched T cells. So that takes place after the enrichment step. And we recommend using a range between 2 to 4 times 10 to the 8 T cells for seeding. For expansion and harvest, so the end product, here we reach a mean of 1.55 times 10 to the 10 T cells after 12 days. So here you can nicely see the range that we were able to generate in 9 runs. Here we had a transduction efficiency with a mean of 39.8%, but this is highly dependent on the construct, the MOI, and the starting material, of course. A mean of CAR T cells were generated of 6.5 times 10 to the 9. And the final volume that you get out after cell harvest can be either 100 ml, but it also can be customized. So the software enables to adjust the final volume. And talking about adjustment, so you can easily customize during cultivation phase your activity matrix. So depending on your needs, you can program different activities like a medium bag exchange, a medium exchange, a feed, culture washes, and so on. And you can easily program it according to your needs. How does that look? So in the activity matrix, you will see this on the screen. And the matrix shows the date, time, and the time of the activity and the type of the activity here. Also, there is an auto calculation of the culture volume, and it will be auto sorted. So they will be sorted accordingly to their performance, so in the right order. And then you also can see the parameters of the activity here. And on the side, you can add or delete an activity and also edit activities. Just to show you our standard protocol for the TCT large scale, here you see that the standard protocol is 12 days long. It ends with the end of culture activity, of course, and a volume of 600 ml. After the end of culture activity, as I just said in my previous slides, you have either 100 ml or different volumes according to your needs, up to 600 ml as possible. And throughout the culture, here you see that several media exchanges are performed and the volume is constantly increased to also ensure that the cells are able to expand accordingly. Here, I just want to show you a little bit of data. So here you see T cell isolation data and cell frequencies on day 0 after enrichment and on day 12. And you can see that with our CD4 and CD8 T cell isolation, it is possible to reduce contaminating cells such as granulocytes, monocytes, and B cells. And we get sufficient frequencies of T cells to really ensure a nice T cell expansion. For our activation, I said it also before, we have our max GMP T cell transact large scale, which really ensures a robust activation of our T cells. This is provided in a vial and can be easily spiked onto the CliniMACS Prodigy. It enables a physiological and efficient stimulation, and it is volumetrically dosed. And in this dot plot, you can also nicely see that T cell activation markers are nicely expressed after the usage of our T cell transact large scale. For the expansion phase, here you can see in this graphs the total cell count that was determined throughout the culture. And as you see here, we only had 2 time points where we took samples and measured them. And you can nicely appreciate that the cell count is highly increasing throughout the culture until day 12. And T cells nicely expand. And here they were cultured with Tex-Mex plus IL-7 and IL-15. And the reason also why I show you the slide is because you can nicely see that it doesn't matter whether you use fresh or frozen material or whether you use IL-7 or 15 or IL-2. The cells nicely expand and the viability always stays above 90% here throughout the culture. Lastly, I like to show you that we also offer trainings on site to really learn the whole process in 1 week. And we do that by giving an introduction into our TCT process. This is a 3-day face-to-face training, including theoretical lectures and 2 simulated runs. And now we also then subsequently offer a 2-day face-to-face TCT-LS training on top, which also includes theoretical lectures and practical training. And here you really can understand the process. So in 1 week, you get the whole training and also can become expert in these processes. And we offer more information on our website, so feel free to check it out. And just to summarize what I just showed you here, so I talked about the CliniMACS Prodigy Platform, a fully automated platform, which also holds our max GMP reagents and consumables that are really optimized for our TCT-LS system. The TCT-LS key features are that we have an enlarged cultivation chamber, which enables a generation of up to 20 billion T cells in 12 days. And we have the max GMP T cell transact large scale for sufficient activation and which enables the expansion of T cells. So the benefits are that you can flexibly start with fresh or frozen leukapheresis. The protocols are customizable and adaptable. And what Ian will also talk afterwards is that you also have the potential to reduce your production time because you have this enlarged cultivation capacity. And I would also cover here that or mention that we also offer service and support. So as I said, we have intense hands-on training, technical support, and we also offer our customized application service in case we need to customize the application in a certain way. And with this, I will hand over to Ian.
So thanks very much, Sophia. So now we're going to dive into a bit of science and some particular application of this large-scale manufacturing process. And to start, I'd like to give a little introduction to the therapeutic target that we're targeting, where we're manufacturing cells to therapy AML patients. So the target is the NPM1 neoantigen. So nucleophosphamine 1 itself is a protein that acts as a molecular chaperone. And you can see down at the bottom here, it's normally located in the nucleolus. In the case of the mutated form, this DNPM1, there's a loss of the nucleolar location signal, and you get a cytoplasmic distribution of this protein. You get this deletion of the localization signal here, because right at the C-terminus of the protein, there's a 4 base pair frame shift, which leads to the generation of a neopeptide. And this occurs in about 30 to 35% of AMLs. So it's a really nice neoantigen target, which is quite commonly present in AML patients. We were lucky enough to enter a collaboration with a group at the Leiden University Medical Center who'd isolated TCR specific for this peptide from a healthy donor. We cloned this then into our proprietary Miltenyi lentiviral backbone. And this TCR recognizes a particular peptide at the C-terminus of the protein called, well, clavepsil. And this peptide binds MHC class 2. And interestingly enough, this complex here requires a stabilization via CD8 to have the full functionality. And you can see that in this diagram here, that in CD4 and CD8 T cells, which we isolated separately, you see no killing of the target cells from CD4 T cells modified with the T cell receptor, but CD8 cells modified with the T cell receptor show a good target cell cytolysis. So as we saw, there was a CD8 dependent activation and functionality of the modified T cells, we decided to develop a slightly different process where we don't just isolate, or we don't isolate CD4 and CD8 T cells for manufacturing, but we only isolate CD8 T cells. So we start this process with a frozen apheresis. The apheresis is thawed. We separate then just the CD8 T cells. We activate 2E8 CD8 T cells to start the cultivation process. And on day 1, we perform a lentiviral transduction. And we decided also in this manufacturing to reduce the manufacturing time to 8 days so that we can get a more rapid manufacturing window and then have a fresh drug product we can directly infuse into the patient. But we do have the possibility to extend the process by a further couple of days in case we don't reach the target dose at this time point. But I have to say, in all the development runs and qualification runs we performed, we always managed to reach the cell dose within the 8 days. And during the manufacturing, it's important to take in-process control samples, to observe the process, to make certain decisions during the manufacturing. And just a quick insight into this based on a CAR T cell product, you can see there's a requirement to take these samples out at different time points. First of all, of course, you need to look at the donor or patient eligibility for the trial or the treatment. But once you have taken your blood sample, whether it's an apheresis product or some other blood product, you need to check that the number of cells present in the drug product are adequate for manufacturing. Then once you performed a cell separation, we need to quantify the number of cells we're going to take into culture. So in the case of the process I'm sharing with you, these would be the CD8 cells. We need 2E8 CD8 T cells to start the process. In the case of a CD4, CD8, you could have this as a higher number, for example, 4E8. And then during the manufacturing, after your transduction of the cells, you want to check the transduction efficiency to make sure the transductions occurred efficiently. And at the end of the manufacturing, you need to check the CAR T cell quality, the numbers of CAR T cells you have, the viability, and all the other release criteria that have to be measured and analyzed. And for the CAR T cell world, we've generated some very nice tools here for the analysis of the CAR T cells during the manufacture and also during patient monitoring. These tools can be used. So we've a number of different panels of ready-made recombinant antibodies, which can be available in a dried format, stain express. And these panels can be used to stain your cell products or intermediate controls. And with the help of express modes that run on a Max Quant platform, you can have fully automated acquisition and analysis of your samples. We also have coming soon similar panels for analyzing genetically modified NK cells. So just some examples of the analyses we performed during the manufacturing of our own product. This was using the cellular composition panel. You can see that prior to enrichment, we have a quite varied picture, as you'd expect in different apheresis products. But after enrichment, we have almost only T cells present. So in this case, nearly only CD8 T cells. And in two of these runs, we co-enriched CD4 T cells as well. And as you can see, then the product just contains CD4 and CD8 T cells. What about the other characteristics of the cell after taking them into culture? As you can see, the viability towards the end of the process is very good, near to 100%. At the early time point, you see a little bit more spread of the viability. This is due to the stress the cells have upon freeze thawing and taking into culture, where you do see a little bit of cell death in the first few days. As far as the transduction and expansion of the transduced cells goes, you can see using a specific tetramer to detect our modified T cells, you can see we get a very nice expansion of the modified T cell population, which are almost completely CD8 T cells. So this expansion we did in the TCT-LS medium with IL-7 and IL-15. And at the end, in these development runs, we saw we had a mean of about 8 times 10 to the 9 CD8 T cells, where about half of these were modified with TCRs. So we could show that within these 8-day process, we could generate clinically relevant numbers of TCR-modified T cells. One characteristic of TCRs, which has to be kept in mind, is the fact that the TCR expression does change during the manufacturing. So if you take out different samples and look at the levels of TCR expressed on your T cells, you will see some variation. And this modulation of the T cell receptor is just due to the activation status of the cell. So CD3 and T cell receptor, they'll be upregulated as the cells are activated. And then as the cells cool down, you also see a reduction in the levels of the TCR expression. However, if you look at the number of integrated copies within the whole cell population, you see that the integrated copy number stays very constant. So there's no variation in the genetic modification or loss of modified cells, for example. This is purely due to the expression kinetics, due to the activation of the T cells. And this is an example of the staining you can get at the end of the manufacturing process, where you see about half the cells are expressing the TCR. What about functionality? Well, we tested the functionality by incubating the TCR-modified T cells with the target cell line, either expressing the mutated form of the nucleophosphine or a wild-type form of the nucleophosphine. As you can see, only in the presence of the mutated form do we see a dose-related killing in this in vitro assay, and also production of cytokines. We see no production of cytokines or killing without the presence of the mutated peptide. And we measured these cytokines using our own MaxPlex cytokine detection system, using multiplex beads, which allow you to measure all these cytokines in parallel in one particular assay. We also tested the functionality in an in vivo model. Here, tumor cells were engrafted, which express the mutated form of the nucleophosphine. The mice were randomized just before injecting the T cells, TCR-modified T cells, and then we monitored the tumor growth every 3 to 4 days. And as you can see, after 17 days of tumor growth and just 10 days after injecting the modified T cells, you can see that in the mice that were only inoculated with the tumor or just treated with mock treated T cells, you still see the strong tumor presence, whereas we see quite a good control of the tumor growth using the DNPM1 TCR-specific CD8 T cells. So I hope I've given you a demonstration of kind of real-world data using this TCT-LS system. We've shown that we can manufacture the TCR-modified T cells from a cryopreserved apheresis product using a shortened manufacturing process of just 8 days and a large-scale manufacturing system. And in this, we were able to generate about 4E9 TCR-modified T cells on average with viabilities of over 97%. And these cell products demonstrated target-specific cytotoxicity both in vitro and in vivo. And we're now moving forward with this interesting T cell receptor now into clinical trials. And this will also be published in the next few months as well. It's been already submitted. So thank you very much for your attention, and we'll be happy to take questions now.